Namaste students, I am JVN Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth, Women's University, Jaipur. Today I am going to discuss about the topic thin layer chromatography, that is part of the chromatography separation techniques. So what is that chromatography? Chromatography is a technique which is used for the separation of the particles on the basis of their polarity and their uh, size, whatever different kinds of chromatography exist, such as paper chromatography, thin layer chromatography, affinity chromatography, size exclusion chromatography, gas chromatography, high performance liquid chromatography. So these are all are the part of the chromatography. And today I'm going to discuss about the thin layer chromatography. What is the thin layer chromatography? As the name indicate, the thin layer chromatography is a technique in which we use a thin layer. We are using a thin layer. So in the thin layer chromatography, a thin layer of a stationary phase is used on which our sample is loaded, and it will migrate towards the end, towards another end by the mobile phase. So what is the stationary phase? Stationary phase is a phase on which our sample is loaded. The sample is loaded, which is known as the stationary phase. And what is the mobile phase? Mobile phase is the phase which carries our sample for separation. So the basic thing are station phase and the mobile phase, which will separate the two molecules differently. So one by one, discuss everything. Thin layer chromatography. The first thing is stationary phase. What is the stationary phase in thin layer chromatography? The stationary phase is a solid particle. It is solid. It is not a liquid. So there are different kinds of stationary phase will be there. Gas chromatography, gas, liquid, solid liquid. So different chromatography exists, liquid chromatography, solid liquid chromatography, different type of chromatography exists, which depend on the stationary phase on the mobile phase. So in the thin layer chromatography, the stationary phase is solid, and it is composed of the glass light. It is composed of the silica on a glass slide. Okay, so we apply a gel, silica gel in the glass slide. How? I will explain. Then next, which we can also use is aluminum oxide. Can also use aluminum oxide, aluminum plate, and silica. These can be used as a stationary phase. So how we can prepare a stationary phase in the lab? If we want to prepare a stationary phase in the lab, then we need to have silica gel and distilled water and glass slide. Three things: silica gel, glass slide, and distilled water. First, we need to weigh. First, we need to weigh silica gel. Suppose that we have weighed approximately 20 gram of silica gel. Okay, and according to that silica gel, we need to add 10 ml of water, 10 ml of the distilled water, and then we mix the silica gel and water properly. If they have been properly mixed, then we need to uh, pour the silica gel and water. When they have already mixed, complete viscosity has been achieved, and then we need to transfer the silica on the solid support, that is glass slide. So we carefully make a layer of the silica on the glass slide and allow it to dry. When it has been dried, then we keep it in the oven for 30 minutes approximately, so that it can be dry, it can properly dry. Okay, when it has been completely dried, then we can use it as a stationary phase. So that is the basic of the stationary, how the stationary phase is prepared in the thin layer chromatography. The next is the mobile phase. In the thin layer chromatography, the mobile phase which we use is the organic solvent or some inorganic molecules also. So mobile phase is the phase which carries our solvent. So in thin layer chromatography, we can use some organic solvents and some non-organic. The organic solvents include petroleum ether, thiethyl ether, acetone, acetic acid. So these are the organic solvents which we can use. Similarly, we can also use some non-organic, such as the ethanol, water, hexane, and pentane. So these can also be used in the mobile phase as a solvent. So what is the use of this mobile phase? This mobile phase will use will take the sample in a particular direction for the separation. The basic thing which apply for the separation is size and the uh, polarity. The first thing is the polarity of the organic solvent and the sample must match. If they will match, then they will be separated easily. Okay. The next thing is the size, and the size is very important. The molecule which we want to separate, for example, if we want to separate the protein, then the protein size must be smaller so that it can diffuse inside the silica gel. Okay. Okay. So if it has smaller size and it will diffuse inside the silica gel, then it will be separated fastly. Okay. But if the size of the molecule is larger and it cannot diffuse freely into the silica gel, then it will be harder for the molecule to migrate. So it will migrate, but it will migrate for a few distance. It will not migrate all long distance of the solvent. It will migrate only for the few distance. Two things are there. First, the matching of the solvent to the molecule. If they will match the solvent, if the molecule which we want to separate will dissolve in the solvent, then it will be migrated. It will migrate towards the end. Okay. If it is not soluble in the solvent, it will not migrate for one distance. So it must be soluble. So the molecule must be soluble in the solvent. Second thing is size. The size of the molecule will be larger or smaller. The smaller particles will move faster, but the larger particle will move slower and will not migrate at the end. So these are the basic things. This is the characteristic. What is the next thing? Attraction of the compound to solid or liquid phase is the basic characteristic. How? What is the mechanism? So when we have applied this sample on the stationary phase, first we need to apply we need to apply sample on the stationary phase, and then we put the glass slide inside the solvent, and it will the solvent will migrate and when it has been migrated towards the third fourth of the sample, then take out we need to take out the uh, plate. We need to take out the glass slide. Okay, and we need to dry it, and when it has been dried in the oven, we uh, we spray it with a spraying agent like in hygiene. Then we observe some mm, some dot like structure on the uh, glass slide, on the silica gel glass side, and we take the distance. We measure the distance of the glass slide. If we measure the distance of the solvent, okay, we measure the distance of a particular kind of dot, so that we can analyze the RF value, reflection value. So higher the RF value, higher the rate with which the product has traveled. That RF value is higher, it means the molecule is more soluble in the solvent. Okay, it is more soluble in the solvent. It has more traveled. So we can analyze our sample on the basis of the RF value. So what are the advantages of the thin layer chromatography? Thin layer chromatography is simple. We need only glass slide, silica, and solvents. Three things. So it is simple. It is very cheap because silica, glass slide, and solvent are not very expensive. So it is a cheap method. It is very easy to conduct because we can find everything, and experimentation can be completed very easily, and the recovery of product is very easy. Okay, so these all are the advantage of the thin layer chromatography. What are the disadvantages of the thin layer chromatography? The disadvantage of thin layer chromatography is that the plate length limited separation up to limited length. Okay, we cannot separate a large number of some molecules. We cannot separate large number of particles to thin layer chromatography. Similarly, the sample which is affected by the humidity and temperature, it can be affected by the humidity and temperature. So these are the two disadvantages of the uh, thin layer chromatography. Here I must share the screen so that you can understand. So this is the diagram of thin layer chromatography. Here this is the drop of where we have spotted our sample, and this this is the stationary page on which our sample is loaded, and this is a uh, this is a beaker on which our sample is present, and it is a this is a starting point, and it is the solvent. So the sample will migrate after loading the sample, and then we will observe some spots like this. So these are the spots, and these are the spots. So this is the distance B, which indicate that the solvent has been traveled till here. So this is a distance B when the solvent is traveled uh, in the stationary phase, and this is the distance where our sample have been traveled. Okay, or so if we want to analyze the RF value of this sample, then the distance which is traveled by the solvent is here, and the distance which is traveled by the sample A, sample B, sample C, sample D. If the sample A is traveled till here, then we will measure the distance. For example, it is 1 centimeter, and if the solvent is traveled 8 centimeter, then we can calculate 1 by 8 Then we can calculate the RF value. So RF value is the distance on the spot of the TLC plate and distance of the solvent. So we can analyze the RF value. So I hope the students will better understand what is the basic about the uh, thin layer chromatography. In this lecture, I will discuss about the application and general use of the thin layer chromatography. What is the thin layer chromatography? What is their stationary phase? What is their mobile phase? What is their basic principle? What is their mechanism? And what is their advantage? What are their disadvantages? So each thing is covered in this lecture. What kind of questions can be asked according to this lecture? The question which can arise: explain the vapor chromatography and thin chromatography. Explain their characteristics, their mobile phase, stationary phase, and their mechanism of action. Explain with diagram and how the R value can be calculated. So you can answer very well. So my lecture is over here. Thank you very much. You can ask me anything. Thank you.